silent Michelle, who is sending emails like crazy in the background. Here we go. Fantastic. Well, let's make a start now that everyone is here. People will be dropping in and out throughout the session, but it's wonderful to have you with us today, Thomas. For those of you who may not know, Thomas has a Bachelor in Life Sciences, a Master in Cognitive Neurosciences from the University of Wisconsin. Then he did his PhD in Cognitive Sciences in Paris before he moved to Australia, where he did his postdoc and is currently also still a adjunct research fellow. And in, last year, 2021, we got him back to France as a tenured researcher or charge de recherche at INSERM, where he will also be hosting his lab at the Paris Brain Institute, also known as ICM, at the Petit Salpêtrière Hospital. And today he's here to talk to us about sleep and consciousness and how these two phenomena are not completely separate and tend to intermingle every now and then. Without further ado, the floor is all yours. Thanks for being here.
Well, thanks for having me, and it's a great opportunity for me to talk about my work. So I will try to cover a lot of things, because I'm not expecting you to all to be aware of the latest development in sleep research or mind-wandering research. The idea here is not to dive into too many details, but to cover a bit the topic of my research, and we can dive into details in during the questions or offline after the talk. So I apologize in advance if there is a lot and that prevents me from providing all the necessary details. And as you said, Stephanie, today I wanted to talk about the sleep and how sleep might be a bit more complicated than we used to think, especially how local modulation of your physiological states will impact the way you sleep, but also the way you're awake. So this is really a topic that has been present in all my research career. I started investigating sleep from a neurophysiological perspective through intracranial recordings in epileptic patients. And this early work was kind of the cornerstone of what I did after, because it really showed that from a physiological perspective, sleep could be seen as a local phenomenon, or at least there are some patterns of sleep activities that can be generated locally in the brain. So what I did after that during the PhD, which I did with Sid Quider, was to look at the functional consequences of this local sleep within the night. And in particular, does it allow us to interact with our environment if parts of our brain is not sleeping as deeply as other parts? And this is what I will talk for the first part of the talk. And a bit fed up with sleep research, I decided to do something else when moving in Australia, so to study awake people behaving, doing interesting stuff. And I was in particular interested in the fluctuations of attention and mind wandering. And that was a bit reinforced by my personal experience. After the PhD, I traveled for a year with my wife in a car. And at some point, being a bit dreamy, I was thinking about something else and managed to put the car on the roof. So nothing bad happened to us, but it was a good demonstration that sometimes we can be quite out of touch with reality. And I wanted to investigate what happens in the brain when we are distracted. So it turns out that although I wanted to escape a bit sleep research, it came back to me because my main conclusion is when you're distracted is perhaps because part of your brain is actually falling asleep. So that will be the second, the other side of the coin, how when you're getting tired, part of your brain can transition to sleep-like activity, and how that will impact your ability to interact with your environment, but also the content of your own thoughts. So now, just to give you the full picture, I'm back in Paris indeed, and starting a new team with colleagues here at the Paris Brain Institute. Isabelle Arneuf, who is a professor of medicine, and Delphine Audiette, who is a cognitive researcher. We call it the dream team for the topic we're studying more than necessarily the members. But our aim is to do this joint investigation of sleep, dreaming, and consciousness, so both sleep and wakefulness in basic and clinical research. So it's we are at the early step of setting up the team, but if you're interested in this kind of work, just reach out to us. We're very happy to receive any feedback, comments, or expression of interest. So to introduce this concept of local sleep, I think it's important to remind a bit why local sleep is a new concept and why it's interesting. And the starting point of this research was the classical view of sleep as a local phenomenon. So we know from human recordings and sleep medicine that, of course, in humans, sleep is not an on and off state. There are different depths of sleep. But the idea until very recently is that although it's graded, when you're in a specific depth of sleep, that's something that will affect your whole brain quite homogeneously. So if you're lightly asleep, your whole brain is lightly asleep. If you're perfectly awake, your whole brain is perfectly awake, and so on and so forth. And the transition from wakefulness to deep sleep will be characterized by a slowing down of EEG oscillations. So you will go from fast desynchronized activity visible in the scalp EEG to high amplitude slow wave that are hypersynchronized. And the reason why you have this hypersynchrony at the EEG level is because of the activity of individual neurons, which in sleep start oscillating between state of activity and inactivity. So you have this massive synchronization between on and off state that will generate these slow waves. So obviously, when you're in a state where vast assemblies of neurons, thousands, millions of them are doing the same thing, that will be pretty detrimental to your ability to respond to an external stimulus or to maintain any conscious thoughts. And that's why in deep slow wave sleep, you are usually not aware of your environment and not able to respond. But something that has appeared recently is that it's very rare that actually you are sleeping in this state of complete synchronization with slow waves everywhere. Quite often, you can be in a state where some brain region will do something a bit different than others, and perhaps sometimes will even show wake-like activity where the rest of the brain is showing clear signs of sleep. So these differences from region to region can perhaps facilitate the processing of external information. One of the first evidence for that was obtained in intracranial recordings, where here I show you the activity that you will see, for example, on the scalp. So that's the middle line. And you see these high amplitude slow wave that are so characteristic of sleep. And two additional brain regions were recorded in these patients, one over the prefrontal cortex and one over motor areas. And what you can see that from within the brain, these two brain regions are doing something quite opposite. While the prefrontal cortex is showing something very close to the scalp electrode, so expression of this sleep slow wave, the motor cortex is actually showing a pattern of wake-like activity. So in a normal individual, you wouldn't have access to the intracranial information. So all you will see is the activity of the scalp, and you will conclude that the person is just soundly asleep. But as soon as you're able to record local activity, you can see that actually it's not because the global activity reflects sleep that you don't have local activity that are reflective more of wakefulness. So this is something that can be a bit generalized by looking not at one single participant, but more data. And when we did that with Yuval Nir, using still intracranial data, we found out that this classical picture of sleep as a global phenomenon. So when you see a slow wave on the scalp, you will see the same slow wave everywhere in the brain and it will be associated with neuronal silencing everywhere. This is quite rare. And most of the time you have things like this. For example, a slow wave at the level of the scalp that can be seen in one brain region, but not another. And later on, you can have a slow wave in another brain region associated with this phenomenon of neuronal silencing. But here, the other hemisphere is not showing that slow wave, and you don't see that slow wave on the scalp either. So you have this local generator of slow wave that will sometimes shut off one brain region while others are still talking, or conversely, sometimes you can have a massive shutdown of the brain, but still a brain region that can potentially encode external information. So the question I asked, and this time it's work conducted in healthy participants, so using non-invasive recording, is whether this local modulation of sleep can support cognitive process and covert responses. So is the brain still responsive during sleep, even if you are not behaviorally responsive? So to do that, with SleepReader, we designed a series of paradigms in collaboration also with Tristan and Beckenstein in Cambridge and Louise Goupil. These paradigms were quite simple. The idea was to overtrain healthy individual to do a very simple task. So for example, distinguishing between words referring to object like knife, table, etc., and words referring to animals. So it's a classic semantic discrimination task. You're receiving stimuli one after the other, and you just have to indicate through hand responses whether the stimulus is an object or whether it's an animal. And the task is pretty repetitive. The participants we recruited for this experiment were young students, so generally sleep deprived. We sleep deprived them even a bit more than usual before the experiment. We tested them usually early afternoon in a dark room on a very comfortable reclining chair. So you can imagine that the likelihood they will fall asleep was pretty high. And it's what indeed happened. What they didn't know is as soon as we could see that they had transitioned to a stable state of sleep, and we do that through scalp EEG, we change the list of stimuli they receive. So they still receive the same categories. So from a task perspective, the task set was continuous. But we presented them with new items in order to check whether their brain was still able to process these new items and categorize the semantic category of these stimuli based on the training and the automation that was performed during wakefulness. So to check that they were still categorizing the stimuli, we looked at signs of motor preparation because each stimulus category was associated with a site of response. Obviously, responses, actual button presses in sleep are pretty rare. And we actually excluded any trials where we had a behavioral response to look only at things that will be considered by any sleep researcher on Earth as true sleep. But since they had to prepare for left and right responses, we could look at covert indexes, EEG indexes of responses in the presence of a lateralized readiness potential. So let's say you're awake and you have to do this task. So sometimes you will answer right, sometimes you will answer left. We can compute an index of the preparation of these responses by subtracting right and left activity in the scalp EEG based on the expected size of the response. And when you do that, what you compute is an LRP. This means that the LRP is capturing differences between right and left hemisphere that is task dependent. So that depends on the response you are expected to make. And what we observe classically when computing the LRP is a negativity over motor electrodes that is starting before the onset of the response. That usually characterize the processes associated with the selection and preparation of the motor response. So what we wanted to check is whether this similar motor preparation are conserved during sleep. So we did a first experiment where we used a lexical decision this time, so deciding between words that exist or not. And what you can see that we have an LRP, which means we have an LRP, I mean we have this negativity being observed centered over motor areas after the onset of the stimulus. So the fact that we have a deviation from zero again cannot happen by chance, because we are comparing the right and left activity according to the expected side of response. So if the participants are doing nothing, you don't have any right-left difference, so you have a flat line. So what we see is a negativity that occurs slightly delayed compared to the wake responses. But it's actually not that delayed when you compare to the very slow response people give when they are drowsy. So that's actually the onset of the LRP corresponds more or less to the tail of the distribution when people start falling asleep. So it seems that they do the task while sleeping here in this light form of sleep. They do that a bit slowly, but not well as slowly as they would if they were actually still awake but drowsy. And the presence of the LRP here is interesting because by looking at an index of motor preparation, we are looking at the end of a chain of processes that involve encoding the auditory information, extracting the semantic content of the words, selecting the appropriate response, and preparing the motor plan associated to that response. So we can be pretty sure that the information we provide to participants is processed in a complex fashion, because they cannot do this chain of processing without extracting the meaning of the word they are being presented. So since it was a quite surprising result, we tried to see if we could confirm with other tasks. So we replicated this NAP experiment this time with a semantic decision, so deciding between words and non-words. And what you can see is that we still have the LRP. The LRP is less sustained, which is perhaps because we have less variability, and it's earlier than in the lexical decision task. And this is quite interesting because semantic decision tasks are known to be performed faster than lexical decision tasks. So this notion of task difficulty could be even seen here in sleep, where it's easier for a sleeping person to categorize words and objects compared to words and pseudo words. But all these were NAP experiments where sleep is a bit light. So we wanted to confirm whether this would happen even during night sleep. So we replicated the semantic decision task during the night, and what we saw is that in light sleep we still see this LRP quite at the same time as during the nap, and again over the motor areas. So in light non-REM sleep at least, so when you have this slow wave on the scalp EEG, but they are just not everywhere, it seems that there are moments in which people are able to process external information quite complexly. But this ability is not conserved throughout all sleep. So if you move to deeper non-REM sleep, so when these slow waves are everywhere, or at least in 50% of each time window, we don't see this LRP anymore. And we don't see this LRP in REM sleep, which is the part of sleep that is closer to wake activity in terms of brain activation, and where most of the people spend their dreams. So at some points, we lose this index of motor preparation, which doesn't mean that the participants were not processing at all. They could still process the stimuli, but what we see is that they are not preparing for a motor response. So with this data, we can try to answer some intriguing questions, like why is motor preparation maintained in light non-REM sleep? How they achieve that? To answer that, you can look at how the brain responds to this stimuli while sleeping. And this is a spectral decomposition of this brain response to this auditory stimuli. So you have time from the stimulus onset and different frequencies. And what you can see is that the appearance of the stimulus will first trigger a huge increase in two frequency bands, the delta band and the sigma band. And that corresponds to sleep rhythms that are triggered by the stimulus. So slow wave and sleep spindles that are thought to be protective mechanisms of sleep. So you present an auditory information, a perturbation to sleepers, and the way the brain will react is that it will try to inhibit this perturbation and maintain sleep. So you have this increase in these two markers of sleep, so an increase in sleep depth that is then followed by a decrease in both of these markers of sleep. And this first reaction of increase and then decrease of sleep depth is quite interesting, because if you look at its temporal and spatial distribution, you can see that the first increase, so this reaction to preserve sleep, is mostly distributed over frontal areas, as if the sleeper was trying mostly to not wake up frontal cortices in order to keep the person asleep. But then the second moment of decrease is more over what will be the equivalent of auditory or motor responses. The interesting thing is also the timing. The timing of this decrease in indexes of sleep depths overlap in time and space with the LRP. So it seems that when you're presenting in light non-REM sleep this stimuli to sleepers, you have a two-step process that occurs. A first one, which is an increase in sleep depth over frontal cortices predominantly, that could help sleepers to maintain sleep and not wake up. And then you have a second step of a lightening of sleep that could allow the sleeper to process the information that it received and decide potentially whether or not it's important enough to wake up or not. So you could have this very flexible local regulation of sleep that could allow sleepers to maintain this connection with the environment without perturbing sleep too much. The other question you can try to answer with this data is, if motor preparation can be maintained in light non-REM sleep, why does it disappear at some point? What is the mechanism that will finally disconnect you from your environment? And we think that the mechanism in question is a mechanism that's referred as cortical bistability. So the fact that if you go very deep in sleep, an activation, like receiving an information from the outside, will turn into a strong inhibition. And that's something that you can see in this contrast between light and REM. In both cases, when you present a stimulus, you have an initial activation that is called the P200, and that is modality dependent. We'll start in the auditory cortex if you present a sound. If you're in light non-REM sleep, this activation will be predictive of your ability to respond. So the more you have this P200, the more you will be able to respond to the stimulus. So you have a positive correlation between the P200 and the LRP. When you transition to deep non-REM sleep, this P200 will be followed by a huge negativity here. So it's actually an evoked slow wave, and in terms of neuronal firing, that will correspond to an episode of neuronal silencing. So that's the P200 that's triggering this episode of neuronal silencing. So inhibiting itself in a way, and you have therefore a complete reversal of the relationship between the P200 and the LRP, this time with a negative one. So when you go deep in sleep, an excitation will actually inhibit itself and prevent you from processing further the stimulation. So you have a tipping point at which, and this is most likely regulated by the state of neuromodulation of the brain, but you have a tipping point where the brain will start inhibiting its own responses to external stimuli and then prevent you from processing what's coming from the outside. Another interesting point to understand in this relationship between the sleeping brain and the external environment is what happens in REM sleep. Because in REM sleep, you don't have slow wave, you don't have this phenomenon of cortical bistability. Brain activity is actually surprisingly similar to wakefulness. So if you're not showing this mechanism that prevent you from answering, what is preventing people from answering in REM sleep? They are conscious as well. Most of the time they are dreaming. So if they can process information from the inside, why can't they process information coming from the environment? We made an hypothesis that's precisely this information from the inside that is competing with external stimulation and preventing them from answering to our stimuli. They are too busy processing their dreams in a way, and that will make them inattentive to our external stimulation. But how can we know what is happening inside their head without waking them up and interrupting sleep? To do that, we computed an index of EEG complexity that was used in disorders of consciousness or anesthesia to index the level of consciousness. So we applied this EEG complexity index to the scalp EEG during sleep, and we saw that indeed it seemed to track very well the different states of consciousness within the nights. It's very high in wake, obviously, then it decreases the deeper you go in sleep and goes back in REM sleep. So it seems to track this ability to maintain consciousness within the brain. We then use this complexity index computed on the baseline to try to predict the ability to respond and the size of the LRP. And what we saw is that in wake and sleep, there is a positive correlation between the two. So the more complex your EEG is, the more you're able to respond to external stimulation. But this was completely reversed in REM sleep. So in REM sleep, the opposite happens. The more complexity you have in the baseline, the less you will be able to process external events. And we think that this is related, or at least compatible with the idea that what really prevents you from processing external information in REM sleep is not because your brain can't process that information, it's because it's already processing something else. So we can hypothesize like these two distinctive mechanisms that will prevent you from processing information when you sleep. The first one in non-REM is a process of cortical gating. So the slow wave will disrupt continuous processing of information, preventing information to be shared among cortical region and preventing you from processing this information complexly. In REM sleep, what you have is a form of informational gauging. So the presence of endogenous information will block the processing of exogenous information. Just a note on the fact that so far I presented you how people can covertly respond to stimuli while they sleep, but my colleagues here at the ECM with other researchers around the world show that you can even have overt responses during your sleep. So they use lucid dreamers, so people that are able to signal that they are dreaming. And they use the fact that these lucid dreamers can actually respond physically through contraction of facial muscles. That is less inhibited than hand response when you sleep. So what they did was to take these people, and when they were lucid, so when they were signaling with eye movements that they were dreaming, but aware that they were dreaming, they started asking them questions, sometimes personal questions like, do you watch football? And they had a code prepared in advance to allow people to answer yes, no question to these questions provided during sleep. And what they saw is that you can really have this two-way communication. So you have someone trapped in their dream, aware that they are dreaming, and suddenly they will receive information from the outside, and they will be able to respond to these queries. So we might be at a very exciting time in sleep and dream research, because we might use lucid dreamers to actually investigate what it's like to be dreaming by asking questions directly to these people and having them answer us back about their experience within the dream. We did other studies about how much you can process information during sleep, but I will just point to the studies without diving too much detail. But we studied whether you can orientate your attention. So can you select different source of information during your sleep using, for example, a cocktail party problem? The short answer is yes, you can in light sleep, light non-REM, and REM sleep. And we also studied whether you can learn during sleep, because if you're processing this information, then the immediate question is, does this processing leave a trace? And the answer is a bit more complicated. I will go, unfortunately, very, very quickly on this because I want to talk about the other parts. But to induce learning, we used a very specific form of perceptual learning, which is a learning of white noise. So we repeat snippets of white noise, and we study whether people are able to learn this repetition during sleep, because they know they can do that in wakefulness. We know in wakefulness that it's an automatic form of learning. It's very fast, it's stable in time. So we use this form of automatic learning that is not very useful in your everyday life, but shows a capacity to learn. And we tested that capacity during sleep. So we had a protocol where we were presenting different sets of white noise to people either in wake, REM, or non-REM. And what we saw is something quite strange. When we retest them in the morning to test their performance on the items they heard during the night, we evidenced a form of learning in wakefulness, which was a confirmation of what we knew, but also in REM sleep. And at the beginning of the memory test, there was no advantage for non-REM items. So they were, in terms of performance, pretty much as new items. So no evidence of learning in non-REM sleep, which is okay, fine. You can learn in REM, not non-REM. That was not so surprising. But what was quite weird to see that when we reached the end of the memory test, by the end of the memory test, the new items are no longer new anymore. So they have learned them a bit. But they still had changed performance for the non-REM item. And then you are in a very strange situation in which if you have never heard them in your life. So you have a form of anti-learning that was quite weird to us. So we investigated what happened during the night because we have an EEG index of this learning. And what we saw is that these items were learned in REM sleep, in light non-REM, but then they were forgotten. And that's the graph you can see here. That's average sleep cycle where you go from light to deep non-REM. You can see that when people start the cycle, so when they're in light non-REM, they have this EEG index of learning. So they show a difference between sequences that were repeated and not. So they were learning these items. But as soon as they went deeper in sleep, they started forgetting them. So how that's possible? We think that the answer to that question is neuromodulation. So this form of learning through exposure, through repetition, is supposed to involve Hebbian learning and long-term potentiation. And LTP is very effective when acetylcholine levels are high. And what happens when, well, the way LTP works is that the famous neurons firing together binds together. So you will strengthen a mnesic trace and activation pattern through repetition. Level of acetylcholine are high in wake, REM, and light non-REM, which is a state in which we evidence indeed a form of learning. But when you go in deep non-REM, this level of acetylcholine actually drops. And there are in vitro studies and in vivo studies showing that if you have very low level of acetylcholine, the LTP rule is actually reversing. And you don't have neurons firing together, binds together, but neurons firing together unbind together. So what we think we're seeing with this white noise is just this reversal and effect of a drop of acetylcholine on basic mechanism of synaptic plasticity in the brain. There are states during sleep in which are beneficial for learning, and states in which actually you will tend to forget. Especially early during a sleep cycle, you will potentially reactivate memories, consolidate them, and later you will forget. And this forgetting is quite important to allow your brain to learn anew when you wake up. So now let's transition to wakefulness. So I show you how sometimes during the night you can have this local modulation of sleep depth and how they can promote your ability to respond to external stimuli. Now I will try to show you the reverse. Sometimes during the day, you can be a bit tired, like today at the end of a long day, and how this tiredness can be associated with some brain regions slowing down, showing slow wave that are not that different from sleep slow wave, associated with phenomena of neuronal silencing, and that's how that will impact performance. So this notion of sleep intrusion in wakefulness was first evidenced in animals, where they showed that if you sleep deprive rats, you can start seeing this slowing down of the EEG or LFP here, with the apparition of this high amplitude slow wave that are usually observed only in sleep. And these high amplitude slow wave are associated with a reduction of the firing of neurons. So they really seem to look like a sleep event just happening while the rat is awake, behaving. What's interesting is with this slow wave is that they seem to index quite well the time spent awake. So it's a good index of how tired you are, because they will increase with the time you spend being active. And they are predictive of behavioral errors. So in this paper, the rats were engaged in a task, and they could predict the missed trial by looking at the occurrence of this slow wave in brain regions that were involved with the task. So that was very interesting, later replicated in non-invasive recording in humans, still using sleep deprivation. So we thought, this phenomenon, is it something that's really related to acute sleep deprivation? So if you prevent someone from sleeping, then of course, maybe some things will go bad, and you will start seeing this sleep intrusion. But maybe that's because they have been forced to stay awake too long. Or is it something, on the contrary, that will happen in a normal day? So we tried to answer this question, so with my colleagues in Australia, trying to see if, without any context of sleep deprivation, we could record this sleep intrusion in non-invasive scalp recordings, and whether they will be able to predict not only your behavior, your performance, but also what's going on in your mind from a subjective perspective. So to do that, we use the most boring task you can conceive with the SRT. So stands for the Sustained Attention to Response task. So it's a very simple task. Yeah, it's a go, no go, where in the classical version, the digit version, you're displayed with a series of digits in a random order. And all you have to do is to press a button each time you see a new digit, except if the digit is the digit 3. So you have lots of goes, a few no-goes. So you can imagine that people get carried away and they start responding a lot to the no-go trials. So it's very boring, very repetitive, and people are often quite distracted while doing this SRT. What we did was to interrupt participants from time to time to ask whether they were focusing on the task, whether they were thinking of something else, which we call mind wandering, or whether they were thinking about nothing, which we call mind blanking. So mind blanking can be a concept that is weird to some of you, because it's not something that is always directly experienced. You have to be interrupted at the right moment to realize that you are actually thinking about nothing. But it's quite interesting that by the end of the task, most of the people who were skeptic about the notion of mind blanking actually experienced it themselves. Myself, I was quite skeptic about the concept of mind blanking before doing this kind of task. So this is just a graph showing you a bit how the attention of our participants fluctuate in this task. So I remember again, the task is really boring. So it's quite normal that they spend only half of the time thinking about and being focused on the task. The rest is split between mind wandering and mind blanking. Note also the huge variability from individual in individual, which can be because of true inter-individual differences or just different understanding of what these different states means. Obviously, if you're either mind wandering or mind blanking, that will affect your performance. You will miss more trials, and sometimes you will also respond when you're not supposed to. Interestingly, we could dissociate mind wandering from mind blanking from a reaction time perspective. Mind wandering tended to be characterized by faster reaction times, so a form of impulsivity, while mind blanking was characterized by slower reaction time, so they were a bit more sluggish. In both mind wandering and mind blanking, although mind wandering was associated with a bit of impulsivity, in both cases, there were their arousal was decreased, and we measured that through both the pupil size and subjective ratings of fatigue. So in both cases, when they were either thinking about something else or about nothing, they were feeling tired. So we thought, all of this can be explained by sleep intrusion during the task. So to do that, we actually detected this high amplitude slow wave. So they're not as big as the one you will observe in sleep. They also tend to start a bit faster, but in terms of their spatial distribution and overall waveform, they're quite close to what happened during sleep. And they're way bigger than any other event like ERP in response to a stimulus. So they are large events, mostly happening over frontal cortices, and they correlate quite well with pupil size and vigilance rating, meaning that the more you had slow wave, the smaller the pupil size was, and the more tired participant felt. So it seems to be a good index of fatigue. What was quite interesting to us is that this slow wave are short in time, so they last less than a second, so they can be detected at the trial level. At each trial, we can try to determine whether or not a slow wave occurred. They can also occur in any electrode. So for each electrode, we could also determine whether or not there was a slow wave. So we can use this information about the presence of a slow wave at the trial and electrode level to try to predict behavior. Whether people, for example, missed the trial, or whether they responded when they were not supposed to, so whether they did a false alarm. And when you do this, you can construct a statistical map of your scalp electrodes, showing whether or not the presence of a slow wave was informative to predict behavior. And this is what we get when we investigated different aspects of behavior in this SRT. So the presence of slow wave in frontal electrodes was predictive of false alarms. So when they were showing this sleep intrusion over frontal electrode, they tended to respond to no-go trials more than when it was not the case. We could also predict the missed trials, but this time when slow wave were showing up in posterior electrodes. And reaction time showing a compound of these two effects, with frontal slow wave predictive of an acceleration of reaction times and posterior slow wave slowing down. So not only with the presence of slow wave, you can predict behavior, but you can do that in a region-specific fashion, different region being impacted differently and leading to different behavioral outcome. We could also predict whether or not people were paying attention to the task or to something else or to nothing. Both mind wandering and mind blanking were predicted by more slow wave over frontal electrodes. But when we look at the properties of this slow wave, especially their amplitude and slope, then we see again this frontal posterior divide, with mind wandering being characterized by larger sleep like slow wave in frontal electrodes, and mind blanking with larger slow wave in posterior electrodes. So it's seen that this sleep intrusion could participate to the genesis of these attentional lapses, with frontal slow wave disrupting executive control, which could mean more false alarm, more mind wandering, and posterior slow wave that will potentially disrupt sensory integration with more misses and more mind blanking. So with one simple mechanism, we could explain the different cognitive consequences of fatigue. Now I will only briefly mention the ongoing studies and how this concept of local sleep could inform not only the general population, but also population with attentional disorders. I focus in particular on the ADHD population because they are often presented as a model of dysregulation of arousal. The combining symptoms of hyperactivity and hypoactivity that is so characteristic of ADHD could be explained because on average they are quite tired, and they try to compensate this by boosts of hyperactivity. Very interestingly, the majority of ADHD adults and children report sleep disturbances. And sleep disturbances actually overlap with the cognitive symptoms of ADHD. So our hypothesis was whether ADHD symptoms could be explained by more sleep intrusion in this individual compared to the general population. So are they showing more of this sleep intrusion, potentially also because their sleep is of a lesser quality? So this is an ongoing project. As I said, we reanalyzed a data set from Raymond O'Connell in Trinity College in collaboration with Mark Belgrove at Monash and Alessia, a master's student here in Paris. The data will be completed as soon as we can. So it's a sustained attention task, not so different from the SRT. And what we observe in the ADHD that they become more impulsive in these tasks. So we have a marker of their attentional dysfunction here. And what we could look in this data set is the brain dynamics and whether ADHD participants show signs of being more tired than controls. And we did resting state before and after the CTET. And what you can see is that before the CTET, this ADHD and controls are not so different. But after, ADHD show more slow oscillation power than controls, which could indicate that they tire quicker than controls. Which prompted us during the CTET to measure the sleep intrusion. So we didn't find more slow wave for ADHD, which could be due to some technicalities about the threshold definition of the slow wave, but we found larger events. So it's more that they show necessarily more slow wave, but these slow waves are bigger in ADHD and control, and this is mostly true over frontal electrodes. So overall, and again, this is very preliminary and very brief, but it seemed that the ADHD brain could be a sleepy brain, and that could explain some of the ADHD symptoms. Now, can we do something about it? Can you act on this sleep intrusion through pharmacological manipulation? This is also something we try to test, and it's a preprint that we will put out quite soon, where we used healthy participants and we gave them different drugs, ADHD drugs. So either stimulating drugs, so methylphenidate, atomoxetine that will boost arousal, or citalopram that will actually tend to reduce it. And what we can see is that if you give citalopram, so a drug that is known for side effects in terms of loss of vigilance, we increase the number of sleep-like slow wave, but we didn't really affect them by using methylphenidate of atomoxetine, potentially because we were studying healthy subjects. We didn't have any vigilance issues. And again, what was quite interesting in this data is detecting the slow wave, whatever the drug participants took in each session, was predictive of their behavioral performance, again, in this region-specific fashion, where you have more we have faster reaction time, less misses when slow wave are observed over frontal electrode, and more misses if they are observed in posterior electrode. So it seems again here in this drug trial that slow wave were an important part of in explaining lapses of attention. So moving forward, I hope to better characterize using, for example, intracranial recordings, the neurophysiology of this sleep intrusion, examining their consequences in terms of cognition or subjective experience, find intervention, pharmacological or non-pharmacological, to reduce them and prevent them, but also examine what could be their function. Do they have some utility in the brain? Can they allow us, for example, to conserve energy throughout the day? Thanks for your attention. Sorry if I decided to present a lot and maybe too much studies, but I hope we'll have enough time for questions.
Amazing. Thank you so much. That was a fantastic talk. And I have to say that it's rare that I listen to a talk with so many brain waves on every slide, and I'm absolutely fascinated. So thank you for that. That was absolutely great. Just to let everyone know how this works, we have a team over on YouTube. We have some people here. So for those on YouTube, if you have questions, just write them in the chat and I'll bring them over here. And for everyone here in Zoom, if you want to ask your questions, feel free to just show yourself and ask your question directly. Or if you don't want to do that or can't do that, feel free to write the question in the chat and then I ask for you. And also, if you need to leave in like 10 minutes at 5, don't worry. It's been great having you. Right, the first hand is up. Leah.
Hi, thank you for the talk. I really enjoyed. I was wondering if I was like conscious sleep, like now we'll try to test all these things. No, I'm joking. I was wondering what happened like with aging. So how this balance between regions that are also active during the night and in the different phase, also what can also be related to another pathology. You mentioned but also dementia. And yes, this.
Yeah, it's actually a very relevant question for me, because I was precisely looking at a data set with young and old individuals before coming here. So it's something I'm very interested in, and that we are investigating here at the ECM. So what we know from the older population is that their sleep deteriorates when we get older, and they also develop more and more impairments in daytime vigilance that can be predictive of neurodegenerative diseases. So we want to go to the bottom of that question, and there are two hypotheses. The first one will be that when you age, the boundary between wake and sleep will be less and less clear. So you will do more sleep intrusion during your day and more wake intrusion during your night. And maybe the two actually interact with each other. The poorer the night quality, the more sleep intrusion during the day, and that could explain why you have some cognitive decline, for example. Another possibility, is that when you age, your brain is less able to produce slow wave than a young brain. And that's something we see, for example, in development. Young babies don't produce as clear and big slow wave as toddlers or teenagers or adults. So being able to produce big waves is something that requires a mature brain in a way. So do we have the reverse effect when we age, and we're less and less able to produce large amplitude waves? Which could mean that during the day, you are not making so much of the sleep intrusion, but you are not sleeping that well neither, and then can have consequences on recovering and the brain homeostasis and brain health. So there are these two alternatives, and the data we have so far seems to favor the second. So initially, I will have gone for the first one because it made more sense to me. But to a certain aspect, there are evidence in the literature that the second one could also be true. For example, if you look at attentional capabilities of older people, it's actually quite good, and people in their 60s are better at attentional tasks than people when they are 25. If you need to do a long drive at night, pick the older driver possible, because they are better at resisting sleep deprivation than younger adults. And maybe this ability is actually due to the fact that their brain are less good at synchronizing. So they don't sleep that well, they don't go that deep, but maybe a side effect is that they don't have a lot of this sleep intrusion. And what we see in our data so far, it seems to be a bit like this. So younger adults seem to have more slow wave, very, very delta wave in wakefulness. And older individuals have less, but they seem to have more theta wave. So their wave seems to be a bit faster and therefore less synchronized. So it could be that the effect of aging, of preventing your brain of producing this slow wave all across the board. So you're still slowing down when you're tired, but not as much as a young, healthy brain.
Okay, thank you. Super
but
interesting.
very, very speculative and preliminary so far. So I might just be wrong.
Okay, we will see. Thank you.
Valentina has a question as well.
Hi, thank you for your talk. My question has a backup story. I mean, this channel has an event every Monday morning, which is Neurocino, and there we present a paper that got us excited during the week, and we discuss about the paper. So last Neurocino was on consciousness. And we started to have a talk about what is consciousness in neuroscience. I was wondering, according to the data you presented, what is consciousness? Can we talk about being conscious while sleeping, or being conscious while awake, but showing the slow waves during a task? This is the first question. The second question is, what were the instructions you gave to the participants when asking what were you thinking? Were they telling the truth all the time, or they were trying to lie?
So I will answer the second question because it's easier. So in terms of the question we asked, precisely it was what was in your mind before being interrupted? And they have to answer, I was focusing on the task, I was focusing on something else, or I was focusing on nothing. So they have some time, some temporal window. We say a few seconds before, so they know that we are talking about the moment right before, but we don't control that. And obviously we don't control about whether or not they're providing honest response. But to maximize that, we tell them that it's perfectly normal to stop paying attention to the task. That's actually what we're interested in, this modulation. So they can feel free to say if they were not paying attention. So we hope that they are honest, but obviously there will be always biases. For example, a person with an ADHD diagnosis might have a bit of a bias to say, yes, I was not paying attention, because that's part of what ADHD is. Or you could have person that have biases in the threshold at which they consider I was thinking about something else, because it's always mixture actually. So you can have this. I'm just wondering, I'm still with you? Yeah, okay. Because screen disappeared. I don't know why is the screen sharing disappeared. So you can have different thresholds that participant will put on this different definition, and that is not something obviously we can control, and that might explain why we have so much variability actually in participant response. And I go back to this slide here, you can see that some people were mind wandering 90% of the time, and some people never. So there will be a lot of noisy, and that's why with mind wandering research you need a lot of data typically. And methodologically, we're thinking about ways to improve this probing of subjective experience. Now, for the second question about consciousness and what consciousness is, you're perfectly right in pointing to the fact that this work can make things a bit complicated. Often, consciousness is being defined through sleep. If you look at articles from consciousness researcher like Giulio Tononi, he will define for the lay audience, consciousness as what disappears when you fall in a dreamless sleep and what reappears when you start dreaming. So very early in the sleep literature, there was this notion that you have clean states. When you're awake, you're conscious. When you're in non-REM sleep, usually you're not conscious. And when you're in REM sleep, you are conscious again of something that is not reality. What we start learning is that it's way more complicated than that. Consciousness is graded, and even in a state where you're not dreaming, you're sometimes conscious of something, sometimes of just being aware and being present. And during the days, there are sometimes that you're not really there, and you have this mind blank where if you're being questioned what you're thinking, you have nothing in mind or nothing to answer. So we are moving from a state where we're considering clean levels to something that is more graded. and therefore, to investigate consciousness, what's important is to ask people about the content, what they were having in mind. And when you do that, you can actually see that with brain activity, you can predict pretty well what people had in mind by the level of activation in different brain regions. So there is a debate in the field about which brain regions are key hubs for consciousness, whether it's in the front of the brain, whether in the back of the brain. But we have the tools now to answer this question by asking people and trusting what they report on their subjective experience and trying to match this subjective experience with brain activity. And hopefully, little by little, we'll be able to understand the mechanism that are making our brain sentient.
Thank you.
There is another question from Ma.
Yes, hello, and thank you for the talk. I have one very specific question about the first experiment that you showed to us about the LRPs. Mm-hmm. So in this case, are the responses, like the preparation was correct? So were you just taking into account the correct motor preparation?
So we don't have that information because the LRP is computed on average. To compute the LRP, you take all the trials where a left response was expected, and you will subtract right minus left electrodes, and all the trials where a right response was expected, and you do the reverse subtraction. So it's really a response on average. And what we see here is an ability to prepare for the correct motor response on average across all trials. In theory, it could be possible to go to the single trial level by looking at, for example, using decoding to look at signs of motor preparation at the single trial level. We tried to do that. That works well in wakefulness. The problem is in sleep, I think we don't have enough data to do a proper training, because we don't have that many trials. So you need to use a wake data as a training, and you have so many differences between wake and sleep background EEG that the models are not really, well, this transfer from wake to sleep is not super efficient. So we struggle a bit to do a single trial, and that's why we kept at the trial average level, and that's why this new work of my colleagues here in Paris is so interesting, because since they are measuring an actual behavioral response, it's much easier to do this single trial analysis. So far, they published their work on lucid dreamers, but they replicated this approach in normal sleepers, and they have actually people able to answer during their sleep with facial muscles. So I think that this ability to respond during sleep, we demonstrated that it exists on average, but it would be very interesting to push that, as you said, to the single trial level, to start having this conversation with people while they sleep and interrogating within sleep their experience.
Okay, and I had another question. So in the experiment in which they have to report if they were like paying attention or wine monitoring, and etc., did you observe like a pattern? Like for example, at the beginning of the task, there were more period of focusing the task, and at the end of the task, the opposite. And if there is like always like a transition, like you first pay attention, then mind wandering or mind blanking, and then at some point you go back to pay attention and so on, like in some pattern.
So I actually, this piece of information, then I removed it because I was wondering if it was too much detail. But you can look at the amounts of on-state mind wandering throughout a block. So there were 10 interruptions per blocks. Blocks were about 12 minutes. So again, at the beginning of the block, people are paying attention, and that decrease. Mind wandering increase, mind blanking also slightly. And you can that also across blocks. There were 6 blocks in this task, it was about 1 hour. And same thing, you can see at the beginning of the first block, they pay attention and then they decrease. What's quite interesting is what happens here at the last, at the very end of the block, because you can see that mind wandering increases sharply. And that's quite interesting because they know it's the last block. So we think there's a weird effect here that they know they're almost done. So maybe they get a bit more excited, and that turns into more mind wandering. And I think this shows also as well the importance potentially of motivation, because when this time you look at the end of the task here, they know it's the last block of the task. They just have 10 minutes left, and then suddenly the on state is increasing back again. So you can see how having different incentive could modify the proportion of time you spend in the different states. Then whether or not one lead to another, that's something I wanted to look, because my initial impression that was associated with some theoretical proposal in the literature, that perhaps mind blanking was an intermediary state between on state and mind wandering, or mind wandering and on state. You are caught off in between, so to speak, and that is a mind blanking. But we did some models of looking at how you transition from, and we didn't see anything like this. And it will also mean that mind blanking should be pretty brief. It could happen very quickly, and we are lucky enough to interrupt people at the right moment. And it doesn't look this way. This EEG index of mind blanking were actually there for 5-10 seconds. So it seems it's more stable. And if anything, I will say it's actually the reverse. We have evidence that mind blanking is a state of sleepiness that is enhanced compared to mind wandering. So the more you progress on the task, and you can see it a bit here, the more you will be tired, and the more you will potentially transition to mind blanking. So my personal interpretation, if there is an order between the two, is you start being focused, then you mind wander more and more, and finally you will end up mind blanking because you're so tired that the sleep intuition becomes so large that you're not paying attention to anything, not even something coming from the inside.
Okay, thank you.
There is a couple of more questions if you're still happy to continue.
Yeah. Again, as long as I'm not kicked out of the room, I can stay.
Oh, you should not say that. I have a long list of questions for you. Right, the first hand that was up was Maggie. Do you want to come on and ask a question?
Yeah, thank you so much for the talk. It was really interesting. Something that I was just thinking about is how we know that waste clearance from the CNS is more efficient during sleep. And like that's sort of part of the hypothesis of the glymphatic function of sleep. And I was wondering whether you think that this waste clearance would also be happening during local sleep, or if anyone thinks that it would be related to the purpose of local sleep, or if that would only happen if the whole brain's asleep.
So yeah, that's excellent question. I see there are some sleep experts in the room. That's something I want to explore through this last thing I mentioned about the function. So this sleep intrusion, are there just accidents? Is it a failure of a system, or it's on the contrary a feature of the system that allows us to do something useful? And we know that indeed in deep slow wave sleep, the state of processing slow wave is associated with this tidal wave in the brain that will carry away all the metabolic waste. And this is a personal hypothesis, I don't think that will happen during the day, because I think that this clearance, metabolic clearance function of slow wave sleep actually necessitates to have a global transition to sleep. So I don't think that this sleep intrusion will necessarily help with metabolic clearance, but it could help with slowing down the accumulation of this metabolic waste by having neurons being less active and burning less energy. So more than a recharging mode, I think the sleep intrusion are more like a power saving mode, where you're slowing down the rate at which you burn your resources. But I'm still not fully committed to any conclusion yet. And this is actually something that we want to study with Matthias Pesiklion here at the ECM. So Matthias is studying motivation, and actually showed very interestingly that if you get people to do over and over again the same task, recruiting the same brain region, what you will notice is an increase in metabolic waste at the level of that region. So overusing your brain will just increase, pretty much like if you do too much physical exercise, you will increase lactate, and that can have detrimental consequences on your ability to continue the effort. So the brain will work similarly. And what we want to do is to look using EEG to see if this increased accumulation of metabolic waste is also associated with more of this sleep intrusion. If the two things go hand in hand, and maybe one could be mitigating the other. So sleep intrusion and their occurrence could slow down the rate at which you accumulate this metabolic waste. So to answer and to sum up my answer to your question, I do think that this sleep intrusion might have a function that is related to metabolism, but it's more in slowing down the accumulation of waste than getting rid of it.
Thank you so much.
Thank you for those questions. Ahmed has his hand up. There we go.
Yes, hi. I have a couple of questions, if that's okay. Mm-hmm. So first is a question related to the earlier part of your talk when you were presenting stimuli, auditory stimuli, if I'm not mistaken, mm-hmm. During light sleep, and you showed that there's that kind of a decrease in the amplitude of the delta and sigma frequencies. Yes, right there. This is more of, I don't know if you've done this at any point of your experiments, where basically at the, I believe like 3-second mark, where you have the most decrease in those frequencies. Mm-hmm. What happens if you then probe with another stimulus right at that point? What are the dynamics? How do they change if you've done this at all in your experiment?
No, we have not done this, but others have done this in the context of teaching people things during their sleep. What they noticed is that you can actually form a new association. For example, there was an important paper on, I think they tried to teach to German-speaking people Dutch words. So they pair a German words with a Dutch word. And what they did is that they had a cue triggering this slow wave, and then they present the pair either at the wrong moment, so at the peak of this response, or a bit later. And I don't remember the details of that paper, but they show that the timing matters here. My PhD supervisor, Sid Guider, also did a study where they tried to target, because this slow wave, they can either be triggered by a stimulus or they are spontaneous. So you could also trigger this moment by just looking at the real-time EEG and providing the sensory information at the right time. And it showed that depending on the phase of the slow oscillation, you will actually favor or not the processing. And this makes sense when you think about the effect of the slow wave, because the through of the slow wave is associated with neuronal silencing, and then the peak with neuronal activity. So if you present during the through, then it's unlikely that your neuron will be able to encode that information, and it's much more likely that it will encode the information after that. So timing relative to slow oscillation should be immensely predictive of your ability to process it for ongoing oscillation. And then this is something you can trigger by having a cue and then presenting another information right after.
Right. Thank you. I'll jump to another question if I can, which is about the anti-learning effect that you were mentioning. So you said that specifically when you present, you present during one specific phase, there's a kind of you can block learning of certain stimuli if they're presented during certain phases of sleep. So my question is, what is there an effect if you have already learned something during wakefulness and then you use that same stimulus during that phase? Can you then cause forgetfulness, let's say, on purpose by specifically using it during that phase? And that could have implication for things like PTSD or things like that, I guess.
Yeah. No, it's something we thought about, and maybe we should do it at some point. So when we started this protocol, we thought maybe we'll have a difference between REM and non-REM, because they are quite different physiologically speaking, and we thought maybe we'll just lump non-REM all together without, considering their different depth in non-REM. So unfortunately for us, there are two things we could have done, but we didn't do, which is, as you said, using some of the wake referent to play them in non-REM and in REM and see how an information you have learned in wake is being transformed by presenting it during sleep. And another thing we should have done is to target light and deep non-REM separately. So we didn't do that. So the consequence of presenting everything in non-REM is that, in a way, it could allow us to show that some of the items that are learned in deep non-REM and they're forgotten, because it's the same items. So the items you learn here at the beginning of the sleep cycle are then being forgotten. So it seems that this reversal of the learning can affect memories that have been learned, but there are two things we don't show here. What will happen for memories that have been learned during wakefulness? Will the same kind of forgetting happen? So can you use sleep to forget about things that you want to forget about the past? Kind of a weird, maybe possibly experiment that it could be interesting to try. The other thing that we didn't try is what would happen for something you present exclusively in non-REM. So do you need the first learning to then forget, or can you anti-learn even if you have not any prior learning? So two things we could explore, but I think you're right that there is a potential for this kind of research for a condition like PTSD. Usually when you talk to journalists, or they are very interested with the concept of sleep learning. Can you learn during sleep and so on? I see it a bit limited by the fact that we're very good at learning during wake. So why bother learning during sleep? Usually the effect we have in sleep are super small, and they come with a quite horrible night. So I think we're all better learn during the day and rest during sleep. So the only advantage I see of sleep learning is if you can do something you couldn't do during wake. And that thing could be forgetting about something you are not able to forget. And we already know that sleep might be actually quite important to resolve PTSD and restore the emotional balance. So it's maybe already something we're doing that we could optimize through careful stimulation. And one example I really like of this advantage of sleep learning is a study from a colleague here in Paris, Anat Alzi, who showed that you can condition people during sleep in a way that you cannot during wake. So what she did was to condition sleepers to associate, so it were smokers, to associate the odor of smoke with a nasty odor like rotten fish. And when you do that in wake, nothing much happened. Because people are smart, they know that it's not because you present cigarette and rotten fish at the same time that you start associating the two. But if you do that during sleep, not only they are not aware that you're doing that, but after they will reduce their consumption of cigarettes for 2 weeks. So you have this very weird effect of learning during sleep that is maybe associated to the fact that the sleeping brain is conserving some cognitive function, but still doing that quite differently from a waking brain. So maybe we can sometimes use that at our own advantage without doing weird things that would not be ethical.
Right. That's I mean that's a good point at the end, of course. Okay, I can stop here, or I can ask one more question. Up to you and Stephanie. Can I go? Okay, one more question. The last one is related to the ADHD findings and different drugs that you used. And you specifically showed that citalopram is more, caused an increase in slow waves compared to the others, of course. But this is more of a sort of about implications for comorbidities and for side effects of drugs. So could this, for example, be one of the reasons citalopram is associated with increases in a drowsiness and fatigue, things like that in patients who take it? And could that also mean that patients who are receiving SSRIs for depression could also now start developing seemingly comorbidities that they didn't have before, like attention deficit, for example, not because it's a comorbidity, but as a side effect because of this kind of effect?
We do things that. Then one thing important for the citalopram case here, it was a one shot, so quite different from the ongoing kind of use in depression, for example. But we did find an effect in terms of subjective vigilance. So although citalopram is actually used because it's not supposed to decrease vigilance, in our case it did, and it increased this marker of sleep intrusion. So it could be that some of the side effects of citalopram are actually associated with this modulation of arousal. For full disclosure, this project is not the one we intended to do initially, because that occurred during pandemic. So we wanted to do here a trial where we'll decrease actually noradrenaline and dopamine because we wanted to increase sleepiness. But we couldn't collect the data, so we used what data we had in hand, and so we collaborated with Redmond O'Connell and Paul Dockery on this data set where we had drugs that were going in two directions. Some drugs were increasing arousal, others were decreasing arousal. And when you look at this data, you see if you decrease arousal, then you have more sleep intrusion. But if you increase arousal, you don't see an effect on sleep intrusion. And I think then the question is all about, it doesn't mean that increasing arousal has no effect, or these drugs have no effect on arousal. I think it's a matter of baseline. So if you have a good baseline, it's not like increasing arousal will do anything, but decreasing it might have an effect. So when you then want to extrapolate these results to clinical situation, I think it's very important to consider what is your baseline of your participants, and maybe the effect we saw here of citalopram are modest, but will be amplified in a population like ADHD that already struggle with vigilance. Maybe you're making things worse. So I think it's very, very important in all the things I presented here to consider this notion of baseline. What I showed is part of your inattention could be due to sleep intrusion, but that's true if you considered the cases in which you get tired. It doesn't necessarily mean that what I showed will explain any form of attention, and you can be inattentive moving to the other side of the arousal scale, being hyper-aroused. And in this case, of course, you will have other mechanisms that will predict these types of inattention. So mechanisms are always true, conditionally true. And I think it's very, very important at the individual level to always pay attention to these inter-individual differences.
Right, brilliant. Thank you so much for these very nice answers. Thank you.
Thanks, Ahmed. More questions are coming in. Don't worry, we're going to release you soon. The first question is, would it be possible to train people to respond more to stimuli during light non-REM, or the other way around, to ignore them completely?
So responding more, I think it's possible. There is this so-called on-call sleep state that has been characterized in medical doctors or first responders population, where if you fall asleep in a situation where you know you will be likely to be woken up, you sleep more lightly. Sleep is actually pretty adaptive. So you might help people answer more just by working on the context, giving incentive. We never tried it, but maybe if we tell people, I will give you more money if you respond more, maybe that will do the trick. There was a very, very interesting study along this line by a Japanese group. So it's Current Biology, published in 2016, where they looked at the first night effect. So the first night effect in sleep research is the fact that if you sleep for the first time in a new environment, your sleep will be a bit different than usual. Usually it's a bit lighter. maybe because you're expecting potential threats or perturbations. And what they show in this paper is this first night effect is has a consequences on the way your two brain hemispheres are sleeping. And more particularly, there is one brain hemisphere, so the left hemisphere, that is sleeping less deeply than the right one, as if one hemisphere was sacrificing itself to maintain a level of vigilance that is higher, and that allows, and they show that through stimulation, that allow them to detect rare tones more efficiently. And that effect disappears the second night. So depending on the context, you might have this ability to modulate your ability to respond. And some people might actually have trouble with that. I have evidence, and that's something I want to test, that some people suffer from what's called sleep state misperception. So they will report very poor sleep quality, but if you record their brain, they seem to sleep normally. So what happens here? My hypothesis is that they actually don't sleep normally. They are very sensitive to external perturbation. They do a lot of local sleep. It's just the way we record sleep in sleep clinic is not detailed enough to show this kind of local awakening. But they participate to this feeling of not sleeping enough, not sleeping well. So there will be inter-individual differences that will make you more likely to answer. Contextual one, maybe motivational one. Whether you can go the other way around and make someone that's responding too much respond less, I think that will be much more difficult. If we had the answer, I think there were lots of sleep troubles that will have been solved by now. And if there is one disappointing thing about sleep medicine, is we know a lot about healthy sleep. We know a lot about disorders, sleep disorders. But sometimes we struggle by improving sleep. There are many people that don't sleep like with the quantity and quality they would like, and we're a bit powerless sometimes to help them.
Right. I would say we cut it here. Thank you so much. Thanks everyone for attending and staying with us. We actually had an amazing attendance of 30 people for an hour and a half. Thank you so much for this talk. If you want to hang around a little bit after everyone is gone, we can have a quick debrief. And to everyone else, have a wonderful rest of the week, and see you for the next talk in March. Bye-bye.
Thanks, everyone.